Good afternoon. My name is Cameron. I'm talking about my PhD project, Interchanging Microbial Communities, the Effects of Soil-to-Soil Transfer. So basically, microbial inoculants, or the addition of microbes, are being proposed as a sustainable or eco-friendly alternative for soil amendments. But before we can give them the big green tick, we need to find out how they respond to being inserted into a novel soil environment. So I don't need to tell a room full of soil scientists and growers how we have a varied soilscape here in Australia. But if you throw on climate variability, different land use management, crop types, you're selecting very different microbiomes. So we're getting a different composition of microbes all over the country. So can you take microbes from a high-performing soil, insert them into an underperforming soil, and make any changes? So to answer this question, I took a curacol from Casino in northern New South Wales, which I'm calling Soil A. That'll be important later. A calcareosol from Hartfield site in South Australia, and a chromosol from southern New South Wales in Wagga. And I used a methodology developed out of the United States called soil-to-soil transfer, which is putting two soils on top of each other, using filter paper as a sort of intermediary to stop the soil particles getting through, and then flushing the microbiome down into that sink soil. Now that sink soil was sterilised with 50 kilograms of gamma radiation because I wanted to give the microbiome every chance of establishing in that sink soil, so I didn't want it having to compete with the indigenous microbes. I used every soil as a source soil, and I used every soil as a sink soil, so it created a bunch of treatments and also some baselines and some controls. So basically the questions that I was trying to answer was, what effect does this transfer method have on the microbial composition? Does this composition, is it shaped by the sink or the source soils? And then do the metabolites, the secondary byproducts of these microbes and the function in the soil correspond to these, either the treatments, the sink or the source microbiomes? So my results show here in the bacterial composition, I did a PCA. If you look at it from a perspective of just the sink soil A, so the curacol, I used all the different source soils as a microbiome transfer. The red group over there is the control measure. So the green and the blue are my treatments. So there's a shift away from the baseline or control measures in terms of microbial composition. Then I wanted to know whether there was an effect of sink and source soils on these compositions. So if you look at the R-squared values, that's a sort of a percentage of how much the composition can be explained by either the sink soil or the source soil. And so roughly between 20 and 35% can be explained, and then pretty significantly as well. And then I wanted to know whether the metabolites and the function of the soil corresponds to either the sink, the source, or the treatment microbiomes. And so what I found is if you superimpose two PCAs over the top of them and then slowly rotate one of them to try and match up, that's called Procrustes rotation. So here we have the different treatments, and what we're finding is you don't have to rotate the PCA very far to get the metabolites to match up with the composition. The Procrustes rotation statistic is, so it's 91% of the metabolites being produced can be explained by the microbial composition. The Mantell statistic shows whether it's positively or negatively correlated, so a value between -1 and 1. So roughly, fairly positively correlated metabolites to the compositions in the treatments. This was also backed up by carbon substrate utilization, which showed a very similar result. So in summary, you're getting shifts through this soil-to-soil transfer method. We're shifting away from the control measures or the sink or source microbiomes, but however, they are shaped roughly by the sink and source soils. The metabolites and the function of that soil is actually corresponding to these sort of novel and new microbiomes that are forming. I didn't have time to go in today, but for the grower groups in the room, I do have plant physiology data and how a wheat crop responded to these new microbiomes. Thank you.